I'm delighted to step in at the last minute and be able to tell you about deep learning and proteins today. My name is Lucy Colwell. I'm in the chemistry department at Cambridge, but for the last year and a half, I've been on leave at Google Research, where I've been working with Philip, who spoke earlier, and Patrick, who will speak later, in the Google Accelerated Science team. And this is a picture of the kind of equipment that they don't let us anywhere near at Google. So this equipment exists, but I have never seen it. I've seen as much as you have in this picture. But we can use it, which is very exciting. And actually, academics can also use this equipment. There's a program for people to use it. And so I'm part of the Google Accelerated Science team. And the goal of the team is to really partner with the experts and accelerate science. And this is Philip and Patrick's team. And it's a very exciting place to be. And I wanted to say at the very start of this talk, in case anybody was interested, that we have a couple of programs at Google for young people, students, postdocs to come and spend time. So one of them is the AI residency program. This is a 12-month research training program. So people are often, they've done PhDs or they've done an undergraduate degree and they want to come and try doing research at Google for a year. And I'll actually talk about the research done by a current resident later in this talk. And we're accepting applications now, so you should, if you're interested, there's a web link here. Just search for AI residency, and you'll find it. And we also have an internship program. The Google Accelerated Science team accepts interns. This is in California, generally during the summer. And if you just look for Google Research intern on Google, or if you go to this little short link, you can find out lots of information about how to apply for that program. So I wanted to get that in before I get stuck into the science. So I'm interested in proteins. I'm going to tell you some sort of work that I've done over the last few years building models of proteins from data, and then some more recent work that I've been doing at Google. So I'm really interested in trying to build models of proteins using sequence data. So we have access to loads and loads of sequence data. You hear all the time about high-throughput sequencing, how we're able to get more and more sequences. And of course, the question is, what can we do with all of that information? How can we use that to actually build models that are useful? And this is something people have worked on in bioinformatics for many, many years. Right? So in some ways, bioinformatics has been using machine learning for at least 40 or 50 years. And essentially, there's a long-standing challenge, which is to find some kind of probability model for the sequence of amino acids that corresponds to a particular protein. So here I've put up the first few amino acids of a protein called hemoglobin. We all have hemoglobin floating around in our blood. And you might ask, what strings of amino acids will correspond to a functional hemoglobin molecule? Right? There are mutations that can occur. We don't have identical genomes. So there are differences between sequences in me and the sequences in you, and also between us and other species and so on and so forth. So there are some set of sequences, all of which will end up producing a hemoglobin molecule. Right? We'd like to know something about that set, and what we have to help us with that challenge is this large set of sequences. So I can go into the database right now and pull out 10, 20,000 different hemoglobin sequences. So I have a bunch of data to work with. And here are some of those sequences. You can see that these different species all have hemoglobin sequences that are quite similar. I've colored these letters, which are amino acids, just by similarity, by chemical similarity. So the red ones, for example, have a negative charge. And we have all of this data. The sequences are very similar. They're not exactly the same. There are changes that occur, and we'd like to use those changes to parameterize models to generate those sequences. Because then we could find new sequences that work, say, as hemoglobin or as any other protein that you might be interested in. And this is also interesting because essentially there's lots of properties of proteins that are very interesting that we'd like to be able to engineer and control that are encoded in these sequences. Right? So it's like there's somehow a code, the code for an enzyme with a particular reaction kinetics or a protein with particular interaction specificity is all contained in these amino acids, and we'd really like to be able to design proteins from scratch. If we could make new proteins, and we could sort of invent machines or nanomachines, we'd be able to sort of make significant progress. And we'd also be able to understand the impact of mutations that occur in genomes. Right? So your genome is different to my genome, and that has consequences, and we don't understand what those consequences are. And if we could solve this problem, then we could make progress with that question. So we can think about this as sequences recording the outcomes of millions of evolutionary experiments, right? So it's like lots of changes get made at random, and some of those changes don't make much difference, and other changes have a large effect, right? And at sort of the very sort of extreme end, some changes actually stop an organism from being able to survive. So what we see is all the successful sequences, all the sequences that work well enough that an organism can consider, can continue to survive. And so we want to be able to take all of that data and invert it to build models that reveal these constraints. So this is another example of an inverse problem, which we heard about earlier today from Cora. And this isn't a new idea in any sense, right? This is a piece from Science in 1973, where Christian Amundsen was sort of gloriously declaring how now we have all of this data, we'll be able to solve this problem. In particular, we'll be able to use all of these sequences to make models that predict protein conformation. So can we be able to predict protein structure? And this certainly seemed like it should work, and lots and lots of people in bioinformatics have worked on this problem, but it's taken a while for this challenge to actually fall. So what makes this problem so hard? We have this unfolded polypeptide sequence. This is a chain of amino acids, and somehow that chain of amino acids self-assembles into a unique 3D structure. So something about the particular order of amino acids in this string encodes this particular folded 3D molecule, right? And this is the problem we'd really like to be able to solve, is to be able to take this particular string and predict the conformation, the atomic coordinates of this 3D structure. And people have tried for a long time to do this with simulations, right? So molecular dynamics in particular has been used extensively. What makes this problem hard? Well, if you look, this is just a very simple set of terms that you'd find in any potential function for MD. There's a huge number of parameters already in these few terms. And this is the simplest type of model that you might make. And more to the point, if we're going to evaluate all of these forces, all of these terms in that potential, and perform integration for every atom in a protein, then we end up having to go round and round this cycle thousands and thousands of times. So if we had something like 50,000 atoms, which you might find in a reasonable sized protein, you're going to have millions of evaluations of this cycle. And so any very small errors in these parameters are going to sort of quickly move away from the correct trajectory. So this is a really hard simulation problem, right? We have all of these atoms. We start with this unfolded chain without any other information. It's really very difficult to find the correct structure. How can we learn from data in this context? So what sort of hints would it be good for us to be able to sort of glean from the data to make more progress? Well, if we knew something about which pairs of amino acids were close to each other in this final 3D structure, then we'd be a lot closer to our goal. And that information is encoded here. This is called a contact map. So this is a matrix, which is all of the sequence positions in this chain against all the sequence positions in the chain. And there's just a 1 for every pair of positions that's close to each other in the 3D structure. Right? So it's a matrix that encodes a lot of information about this structure. If we could get this matrix from the sequence data, then we'd be in much better shape. Right? And this is clearly sort of an easier problem than this 3D configuration, but it'll help us get there. So how might we get information about which pairs of amino acids here are in contact? Well, biologists for a long time have made an interesting observation that turns out to be very helpful. And what they've observed is that often what you'll see in the laboratory is you'll have a wild-type protein that works perfectly well. You'll have a protein with a single change from that wild-type or normal sequence, which is completely broken. It doesn't work at all, right? But then what you find is that you'll have a second mutation at a distant site, a site that's far away in the sequence. But that will rescue the function of the protein. You'll end up with a functional double mutant. And this toy cartoon illustrates one reason why that might occur. Here you had a nice charge pair in this structure that was sort of holding the protein together. The first mutation lost that charge pairing, and the second mutation restored it. Right? So this works perfectly well. It's not exactly the same sequence as before, but it still assembles into the same structure. So what would that mean, that observation, this sort of second site suppressor or second site compensation observation? Well, essentially it's suggesting that mutations at different sites depend on each other. So for a long time in the protein literature, models have been built of protein sequences that use all of this data, but they assume that the mutations at different sites occur independently. That's a big assumption. It's like a model that only has first-order terms, right? So we consider each position in the sequence separately. But this idea is suggesting that actually that approximation is losing a lot of information, right? There are interactions between the positions. So if we were to model those interactions as well, then perhaps we would be able to get more information about the protein and specifically about the 3D structure. And so if we think about this idea of interactions between positions in the protein, what that would mean if we look at many sequences is that we either see the wild type sequence, which in this case had a D and a K, or we see this sequence with a pair of mutations. We see this R and D pair in these two positions. And we never see this intermediate, right? Because it didn't work. So it hasn't survived in the evolutionary record. And what that means if we look at these sequence alignments is that we'll see a correlation literally between these two columns and where they change, right? So it's either D and K or it's R and D. And from a statistical point of view, we're literally just seeing that these columns, numbers 1 and 6 here, change at the same place. That's a very simple observation. We're just going to look at those correlations. Of course, what we'd like to be able to do is, and this is here in the real data, the hemoglobin sequences I showed you can start to see that actually there are some sort of correlations between where the changes happen in these columns. What we want to be able to do, of course, is look at correlations because that's easy and cheap, and be able to use that to make predictions about contacts in 3D structure. So we'd like to exploit the correlations that we see in the data and use them to build a model that can predict the structure. Now that all sounds very sort of nice and aspirational, but what happens when we actually try this? We take a large set of sequences, we're going to compute essentially a covariance matrix, and we'll look at the off-diagonal entries in this covariance matrix, and we'll look for the largest entries, right? So we're going to use something like mutual information to compute a whole matrix of scores, and basically we're going to ask, are the highest scoring pairs in our matrix, do they correspond to contacts in 3D structure? And this is a question that Perutz asked many years ago, back in the 80s or 70s. Many people have sort of thought about this problem hard and worked on it. And of course, now that I've told you that, you know that it isn't that simple, right? So here's the answer. Here's the contact map. That's what we'd like. This is the structural information we'd like to predict. And if I carry out that exercise, I compute covariance matrix of a large set of sequences, what I find is that I get high-scoring positions that are sort of close to the diagonal if I just look at the top 150 or so. But if I start to put more points on that plot, I sort of see these large splodges of red, and the red doesn't do a good job of overlapping the blue, right? And if I could predict accurately, then the red point should really do a nice job of finding these blue contacts, and it doesn't work very well. So why doesn't this work? You see you have this sort of, sets of points that accumulate in certain parts of this map, and there are other areas where we totally miss the points that we need to find in order to know about structure. So what I'm going to allege is that there's noise that confounds this correlation signal. And if we go back to this set of sequences I showed you earlier, what I didn't say at the time is that I'd sort of colored these top ones blue and the bottom ones red. And the reason I did that is because the red sequences are from species that are more closely related to one another than the blue sequences. There's structure in this data, right? What that means is that the red sequences are going to be more similar to each other, not because functionally they need to be, but simply because they're from species that are more closely related. They've had less time to become different. So it's like there's sort of batches in the data. And of course, your sort of graph that we will associate with this structure is this phylogenetic tree, which tells us about the relationship between these sequences. So here, number 8 and 7 are closer to each other. They're going to be more similar than sequence 8 and sequence 1. And so that means that this data isn't IID, right? We have this huge sort of structural artifact in this data, and maybe that's what's causing the issue with our simple correlation analysis. So of course, what you might ask is, well, okay, if I just look at phylogeny, I'm just going to look at the relationship between the sequences, I'm going to carry out a simulation where there's no, interactions between positions, only covariance is going to come from phylogeny, then I see that I end up with a strong correlation structure in my sequences, and I need to figure out how to deal with that. And it turns out that actually you can build a nice mathematical model to help you deal with this. You start by thinking about very simple branching events, and this tells you something about the eigenvalues of the covariance matrices you expect to see, and you can progressively make your tree deeper and deeper. So these matrices get larger and larger. They have this nice block structure where you get higher and higher powers of this parameter alpha. And essentially what you find out is that the phylogeny causes a power law in the eigenvalues of your covariance matrix. And that's nice because even though I'm talking about the covariance matrix sort of between the sequences, if I then look at the covariance matrix between positions in the sequence, it has the same eigenvalues. Right? These two matrices are very closely related, and so it means that if I know this power law comes from phylogeny, then I can get rid of the effects of phylogeny by removing the power law. And so if you carry out simulations, you see that this is indeed true, that these eigenvalues of this covariance matrix follow this power law. If you look at data, real sequence alignments, you'll discover that actually also they have a power law in the eigenvalues of their covariance matrix. And so you can sort of isolate the part of that signal that's caused by phylogeny and essentially get rid of it. And you see that if you do that, you can do a much better job of using the covariance matrix to predict contacts in the 3D structure. So that's great. It means that essentially if we invert the covariance matrix, we can actually do a really nice job of finding these contacts in the 3D structure just by analyzing the covariance of those sequences. So this was a result that we discovered some time ago, and we didn't exactly understand at that point why it worked. I think we now understand that more clearly now. But of course, the question is, was that enough information to actually tell you all about the structure? Right? All that we know at this point is that we have these contacts in the 3D structure. And of course, what we have to do is take this unfolded polypeptide chain and literally enforce these constraints. So in this top right-hand corner, I've used this, I'm calling it a global model, to identify these contacts. I'm then going to enforce them and fold up this 3D chain. And it turns out there's some nice simulation software that you can use to do that, using some fairly simple approaches. And then if you do that, you actually can do a fairly good job with that information extracted from those large sequence alignments of predicting the structure. So here, I've overlaid the true crystal structure, shown in gray, with the predicted structure shown in purple. And these aren't perfect, but they're getting a lot closer to an accurate 3D structure. And of course, you may well have heard that recently DeepMind has made a significant advance, I would say, on this problem. And essentially, they've used an approach that's very similar to this, and they've done a much better job of learning how to go from that large sequence alignment to those predicted contact maps or distance matrices. And they've used neural networks to be able to sort of turn it into a supervised problem and transfer information to proteins where there aren't lots of sequences. So these are some of the transmembrane proteins that we were able to predict back a few years ago. And what was exciting is that we actually predicted structures which have now been experimentally solved and showed that our predictions were within a few angstroms RMSD from the actual structure. So that's an interesting sort of illustration of how we can use those families of protein sequences. But what I want to talk about now is how we can actually model this sequence space. So everything I talked about just now was modeling sequences within a family. Like I talked about hemoglobin, that was a particular set of sequences. But of course, we have to put those sequences into families in the first place before we can carry on and do that sort of more advanced modeling. And the way that this has been done for a really long time is using hidden Markov models, which are actually surprisingly powerful, simple machine learning models that treat every site in the protein sequence as evolving independently of the other sites in the sequence. But now that we sort of have access to significantly more sequences and computational power, it raises the question of whether we can use deep neural networks to do an improved job of the sequence classification task. And the reason that we might want to do this is something like 30% of bacterial protein sequences are completely unannotated. We have no idea what those sequences do. So we have all of these sequences sitting there in the database, and we literally don't know what they mean. So we'd really like to be able to classify new sequences. We have many, many domains of unknown function that we can't annotate, and we're sort of also interested in learning good generic representations of protein sequences. So in image classification, sort of tasks like ImageNet that are sort of practice tasks, if you like, the community has trained models on and got better and better at, have really helped us understand how to build good models of images, and we'd like to do something very similar for protein sequences. So how does this work? Essentially, you have a whole bunch of amino acid sequences. They're all different, and you'd literally like to assign them into classes or protein family. And at the moment, protein families are derived from sequence similarity. So we look at sequences, and we have a sort of metric for figuring out how similar they are, and we use that to classify them. And it's essentially highly informed by something called multiple sequence alignment, which is where we literally sort of line sequences up with each other. And this is an example here. You can see that there are these sort of gap characters introduced, and that corresponds to there being an insertion or deletion event between two proteins. But this is not a completely simple exercise. And in particular, much of the data that we use in the community comes from a resource like this one, which is called PFAM, which is a really rich set of annotations of protein sequences. And it's made starting from a set of seed family alignments that have been intensely manually curated. Right? So we have a whole bunch of knowledge that's been encoded in this set of 17,929 classes, and that's used to train hidden Markov models, which are then used to classify proteins. So the question is, do we have to sort of continue using those same hidden Markov model approaches, or can we develop new approaches using deep learning? So the sequences are between 50 and 1,000 amino acids long. There are something like 1 million sequences in the total seed database, and there's roughly 18,000 classes, 17,929 classes. So you have this huge classification problem, right? There's relatively little data in a sense. 1 million sequences for 18,000 classes is not that much. And what's interesting about this problem is that there's a huge tail of very, very, very small classes. So there's many, many classes, at least in the seed, have less than 10 sequences, for example. They're very small. So all of these classes here have very, very few members in the family, and there's a few big classes sort of towards the end. So essentially, we're going to use this set of seed sequences because there is some level of human verification involved in annotating them, and they're chosen as exemplars of the family. So as I said, at the moment, they're classified using HMMs, and this is a hidden Markov model logo, so it sort of shows you there are some positions where there are particular amino acids that are preferred, and other positions in a particular family where there's more variation. And basically, the way that this model works is that you have 17,929 models. So I have all these different models. I'm going to take a single sequence and sort of try fitting it against each model separately and pick the one that has the best score. And that's a little bit sort of different to the way that deep learning models try to work, because there's a lack of shared model parameters across the families, right? We're literally considering each one separately. And so there's a sense in which it seems like we could, extract more information from this data. And it also relies on this alignment technique. So we're going to try and rephrase this problem slightly using ideas from computer vision. So certainly image classification is very well understood. So we're going to think about domain classification as an image classification problem. We're going to imagine sort of domain calling. So domains are small parts of proteins that occur in families. And so we need to actually sort of segment them from the sequence. So it's a bit like image segmentation, right? Here in this image, we can sort of see that this part of the image contains a dog. And the same way with a sequence, we want to call that a particular part of it contains a particular domain. And this is a project that's really led by a guy called Max Bileski. He's actually in the Cambridge Google office just down the road. We also would like to understand there are different features of proteins, and we'd like to be able to sort of learn representations that tell us more information about those features. a bit like that structure prediction work I told you about just now. So in terms of what metrics we're going to use, accuracy is the sort of most natural metric. We're going to evaluate on the seeds. So we've set up this problem by actually taking that seed data set and splitting each family. We split each family into a trained data set and a test data set. And so we train our models using just 80% of the sequences in each family, and then test whether they can correctly classify the remaining sequences. And part of the reason we do that is we want to be able to compare performance with the hidden Markov model. And so if we retrain all the hidden Markov models with that 80% of each family, it's less clear that they'll perform perfectly at labeling the rest of the data. It means there's some chance for us to be able to actually outperform the hidden Markov models. And so I'm just going to quickly tell you the sort of bottom line results of this exercise. So we've compared a number of different models here. And so we find that even though actually the HMM was used to label the data, because of the way that we split the seeds, so we sort of taken away some of the information that was available to the original HMM when it did the labeling, the HMM isn't quite perfect anymore, and actually our deep convolutional neural network is able to do slightly better than that HMM. I should add at this point that this deep CNN is a ResNet architecture, so that means that it models a residual essentially at each layer. And so we find that the deep CNN, which when we look at different architectures, something like 3 or 4 layers tends to do best, it does do better than this either 2-layer or 1-layer version. And what was particularly surprising to us was that it really did better than the recurrent neural network architecture that we tried. So sequences, when you think about building a neural network for a sequence, I at least think about using an RNN. That's what people use a lot in language modeling. And we found that these ResNets, these deep CNNs, I should also mention we actually used dilated convolutions in this deep CNN. This was able to do very well at this task. But these models are all doing pretty well. So this is a relatively straightforward task, and we're now ramping up to try and tackle some more difficult tasks. But what's interesting is the trained model does learn, for example, similarities between the amino acids. So it wasn't given any information about the amino acids. It was working with unaligned sequences, so it doesn't know anything about which amino acids are really related to each other. This is the ground truth Blossom matrix that has a lot of human knowledge encoded in it. And this is derived from our model simply by examining the embeddings. And the labels here are not very helpful on this embedding one, but essentially you can see the same block structure as in that ground truth model, where the scales are slightly different. So we do learn these similarities. We can, of course, also actually examine our networks, our trained models. And here we're using an approach which is called sufficient input subsets, which is developed by an intern in the Google office in Cambridge called Brandon Carter. So Brandon's doing a master's at MIT, and he came to Google to do an internship. And he did this piece of work during his PhD. And essentially what it does is it examines the network, and it asks, why does that network make the decision it does? Right? Why? What input features are particularly required for that network to make the decision that it does? And it operates by identifying a sufficient input subset, a minimal feature subset, whose values alone suffice for the model to reach the same decision. So these are some examples from MNIST. So this is an MNIST 4. And in order to classify these examples correctly, these are the sets of input pixels that the model needs in order to declare that that's a 4 and not something else. So this is a way of actually sort of trying to probe the black box and figure out how it works. And if we do this for our sequence data, we apply the same technique. What's interesting is we find amino acids, these are in different sequences. These are unaligned when the model sees them. And so the model has actually learned some notion of alignment. You can see it's focusing on the same amino acids in different sequences in the same positions in the sequence. So somehow it is learning, like in particular here, we have this nice little motif that the model is focusing on. So it is actually learning something sensible, and in particular, it's also looking across the sequences, sort of focusing just on the beginning or the end. So this gave us confidence. And this is just comparing, this is the hidden Markov model, the HMM logo, and you can see that there are some features in the HMM logo that our deep CNN is also picking up, which suggests that it's learning useful information. And so we can also start to try and ask, well, if we look at making changes in those sequences, what effect do they have? And you can see that there are sort of features I've marked here, which the model didn't know about, but which have kind of patterns of changes. This is very early work, but it suggests, again, that the model is learning something sensible from all of this data. So the last thing I want to mention in this section of the talk is this sort of domain calling task. So if we look at our whole sequence, can we sort of divide it up into pieces that correspond to different domains? And this is the work by Theo, who's an AI resident. And you can see that actually we're doing a pretty good job of this. So here, we've sort of shown in the score for each of the domain annotations in this whole sequence. This is a whole amino acid sequence that's roughly 750 amino acids long. This is the ground truth annotations that we've gotten from the database, and these are the annotations that our model is calling. It's calling for every single residue separately, and you can see it's doing a pretty good job of identifying the correct annotation for each position in that sequence. And there's another couple of examples showing that here, and so that's very exciting. And so finally, in the last few minutes, I want to talk about finding new sequences. So I've talked about building models for existing sequences where we have annotations. I've talked about predicting structure. What about finding new sequences? Can we really solve this design task? I'm going to talk about this in the context of gene therapy. Gene therapy is now being used to treat and cure chronic genetic diseases, something like 6,000 single gene disorders, many of which have no good treatment options. So the idea here is that you take a piece of DNA, a gene, and you encapsulate it in a capsid, a protein capsid, and you then insert that into a person, and that gene gets delivered to the correct target cell. And it's that capsid that I'm going to focus on in the next few minutes. So one capsid that people used is from a virus called adeno-associated virus, or AAV. So these are little sort of 60-mer regular shapes made out of a particular protein, and we'd like to be able to program these capsids so that we can improve their ability to evade your immune system and efficiently localize them to particular cell types. Right? I'd like to be able to direct them to, say, cardiac muscle or skeletal muscle, or the blood, or any particular type of organ, for example. And so essentially, these little capsids are here labeled in blue, and that's what we're going to be sort of working with in the next few slides. So how does this work? This is a genome map of AAV, and it's this capsid gene, CAP, that we'll focus on here. This gene results in a protein that's about 700 amino acids long, 735 amino acids, I think, to be precise. And essentially, that single monomer forms a very large complex with using 60 copies of that single monomer. This assembles, and it forms a sort of integral complex which packages a copy of the genome that was used to make that protein. So this is really quite a large object. It's about 25 nanometers, and it's formed from 60 copies of this single protein. And you get this beautiful, highly symmetric assembled capsid, and that acts as a container in gene therapy. So we're going to basically ask, if we take lots and lots and lots and lots of different versions of this capsid gene, which ones actually work, right? Which ones are able to sort of, form a viable capsid and don't fall apart? And so, I think I went the wrong way there, sorry. And so essentially what we're going to do is build lots and lots and lots of different versions of the sequence, which are called mutants, and test them. And we'll start off by making single mutants, and then we'll make multiple mutants, first within sort of small regions, and eventually by combining those small regions with each other. We're going to work by dividing this sequence up into small tiles, and essentially the way that you test these sequences is that you make them in the lab. Well, I didn't make them because I didn't know how to do this, but this is in collaboration with a lab actually here at Harvard, who they're highly skilled in making these sequences. And so you take this piece that you've altered, which I'll call an oligo, you then use it to replace the native sequence in a plasmid, and then you then express that plasmid in bacteria. So you make lots and lots of versions of this protein. Something like 10,000 or 100,000 variants can be made in a single library, where these are all the different variants shown in orange. So we're going to make thousands and thousands of different mutants. Which ones should we make? We'll start off by making single mutants, so we'll make single changes. We're going to allow deletions, insertions, what we call substitutions. So substitutions is where I just change the amino acid, right? And I've shown an example of a substitution here. So I changed what used to be an E to a D in the sequence. And there's another example here where I made an insertion. I literally added an extra amino acid into that sequence, right? So I really can just change the sequence however I want. So the first step was to make all the single insertions and substitutions across the entire protein. That's like 30,000 unique sequences. That's already a fairly big experimental effort. And our collaborators then tested these sequences to find out whether they work or not, whether they package correctly to make a nice capsid. And we're then going to use that data to choose multiple mutants to construct, to make more complicated variants. And so this is the data from that single mutant study. Right? We've made all of these changes, and you can sort of see that there are regions. These are the positions in this section of the sequence called TAL21. There's 28 positions here. There's a region here where whatever you do, it seems really bad. Right? So here, red through to white are changes that either make it better or it still works well. And what we want to avoid are these blue changes that break the capsid. And so you can see that there are sort of regions where any change that you make really breaks the capsid. And we're going to start off with a very simple model where we design variants or multiple mutants by combining mutations. And we'll obviously combine the ones that have good single-site outcomes. And the most naive model is to just assume that the mutations add. Right? I mentioned those hidden Markov models. They assume the sites change independently, and that means that you can just add the effects together. Right? If they don't interact, then it should just be additive. So this is like our null model here. And so if we do that, if we use an additive model and we design multiple mutants, then we came up with a whole bunch of sequences, some of which were close to wild type, and some of which were quite a lot further away. And so here I've sort of split out the variants by how many changes they had, right? So we had quite a lot with 2 changes, but we also had a significant number with 6 or more than 7, and in fact as many as 49 changes from that original sequence. So we're getting further and further away. And this is a huge space, right? Because at each place in the sequence, we're choosing within this 28 amino acid region, but there's 20 different amino acids. There's a huge number of different sequences that we could choose. And so, even with all of that single-site data, we're actually finding it difficult to find viable mutations that are further away from wild type, right? So the way that this plot works is I plotted precision on the y-axis. So here, I made, in total, this many mutations, where the number is shown here on this side of the y-axis. And of all of those variants, so of all 5,000 variants that we made, most of the ones that were 2 mutations away from wild type worked. So those are shown in green, and the ones that don't work were shown in purple. And the proportion, so proportion of green as a fraction of the whole, is what's shown with this gray line. So it's like 90%. So sequences that are just 2 steps away from wild type, we're doing a fairly good job of finding, right? We found 90% of them, 90% of the ones that we chose worked. On the other hand, by the time we've got to being 7 to 10 steps away from wild type, we're down below 60%. So we're doing we're being much less successful at choosing good sequences. So this was the result without any kind of machine learning, just using our naive additive model. And so of course the question is, can we learn better models to choose multi-mutants successfully? So can we raise this line? And what we're going to do in order to train these models is work with a set that we've chosen completely randomly. So we're actually going to restrict ourselves to just the single and double mutants shown here. In fact, you can see that if we choose randomly, actually, it's very, very, very difficult to find sequences that are far away from wild type. So out of all the 2,000 sigmas that we chose here, only 29 worked. So it's very difficult. So we're going to train models using this data, and we're going to test them using our design set that we designed using that additive hypothesis. And it turns out that, yes, if we do that, this was our original no machine learning line. It turns out that even actually logistic regression already does a better job at lifting that line. But if we train either a CNN or an RNN on that random data, then we can push that line up even further, in particular in this region where we're sort of 7 or 10 mutations away from wild type. I think what's particularly interesting is that we actually show that we can do that even if we just train on this small set of random double mutants. So it suggests that we're actually able to learn these models with relatively small amounts of data. So that's finding new sequences. So thank you very much for listening. I'd be delighted to take any questions. Thanks.
Questions? Brian.
In your protein folding work, one of the challenges to physics-based models of folding is that lots of polypeptides don't actually fold up spontaneously. They require something in the environment, a chaperone molecule or whatever.
Yeah.
Can your machine learning technique avoid that problem or overcome it?
Well, it depends a bit, but as long as there's signal in the sequence alignment, right? So even though they need chaperones to fold up, you can imagine that there's still correlations, so correlated substitutions that occur. And as long as that's the case, then we can find this information, then it seems like we can avoid that problem. And it'll tell us, sort of what constraints there are, which pairs need to be next to each other, and that helps us get to the answer. So it does seem to help with that problem, yeah. Yeah. Yeah.
Yes.
Your number of 2K of your training set is really, to me, pretty remarkable. If you look at those sequences, are they doing something obvious, like just selecting out the ones that are in contact regions or something like that?
So what was surprising actually is that, so I showed you 2K. I can choose different sets of 2K, and it seems to work equally well. So I was really surprised by that. They were really chosen randomly, and we have we have a larger set that we can choose from, and we can try different subsamplings. So it doesn't actually, we need to do more work on this because it would be a really interesting result if it's true. But yeah, it seems to be that you just need a small amount of information about these doubles. Yeah, no, I was really surprised by that too. So yeah, it'll be interesting if it holds up.
Yes, one more over there.
Could you talk about the correlation in order to figure out which amino acids connect with which other amino acids? It doesn't seem like you're taking into account that some are positively charged and some are negatively charged, and there are certain physics reasons why some would not care about being close to each other and some would.
Yeah.
Wouldn't that help?
Yeah, so you would have thought so. I would have thought so. Some people have tried to include that kind of information, and no one has figured out how to make it really work better yet. But one thing I think would be really interesting is I mentioned the sort of the DeepMind work, and that hasn't been published yet, so it's a result that just was revealed in the recent CAS competition in December. If we were to analyze their network and understand if it's somehow learning something about the different amino acid types, I think that would be really interesting. But at least as far as I know, nobody has managed to actually sort of put it in to the simpler methods, although people have looked at that. Yeah, that's a really good point. Thanks.
Okay, well, if there's no more questions, I think I would like to thank the speaker again.
Thank you.
And,